In this video, I'm going to be going over some of the answers from the biochemistry test review. So first question, for each of the following, what is the monomer and polymer? So for carbohydrates, the monomer is the monosaccharide, and the polymer is the polysaccharide. Monosaccharides are also known as simple sugars. The general shape of the monomer for a carbohydrate is a hexagon or a pentagon. So there are these little carbon rings. And then for the polymer, we get a bunch of these hexagons or pentagons linked together like a chain of these shapes. For proteins, the monomer is the amino acid, and the polymer is the polypeptide. Okay, drawing amino acids. There are two ways to draw amino acids. We're always going to have a carbon with a hydrogen on one side and the R group on the other side, and then another carbon. So this part will stay the same. Just draw that structure. But then depending on whether or not this is ionized, meaning whether or not it is dissolved in water, you get a little bit different structure on the other sides. The sort of simpler way to draw it, the non-ionized form, is like this. And then the ionized form is going to look like this. So you could potentially see either of these, but either way, this part is going to be called the carboxyl group. This part is going to be the amine or amino group. This will be the R group. So those are the parts that you would need to label. Now if I was trying to draw another amino acid, let's just deal with this one right here that I labeled. Bonding with this one, just the important thing to remember is that the amino side of one bonds with the carboxyl side of the other. So you don't want to line up two amino groups together or two carboxyl groups together, because when these bond, we're going to take this OH and this H out as water and then be left with those bonded together with a bond between that carbon and that nitrogen. Sorry, that got a little bit messy there. Okay, types of carbohydrates. Sugars. Short chain, just one or two. Function, quick energy. This is the energy that you would like immediately burn off when you're exercising. There's sugar in your blood, you know, your blood sugar. So this is your first immediate source of energy. Starches are a medium chain, and they are energy storage for plants. So starch can be broken down back into sugars and then used for energy, but it can be stored in this starch molecule within the plant. Glycogen, also medium chain. This is energy storage for animals. So found in the liver and muscle tissue. If you're burning off all of the blood sugar, the quick energy you have in your blood, then this glycogen can be broken down into more sugar for you to have for energy. Chitin, long chain structure for animals, specifically invertebrates like insects and shrimp and crabs. It makes up their shells. This is not digestible by humans, so this would not be a source of energy. It's too long. And then cellulose, also a long chain. This is structure for plants. Makes up their cell walls. And again, this is not digestible. It's too long a chain. It's what we would consider fiber. Dehydration, synthesis, and hydrolysis reactions. So the general idea with dehydration synthesis is that we are building macromolecules. We're using energy to do that. So this would be an endergonic reaction. This is construction of molecules that you need, proteins, DNA, whatever is needed for the body. And then the hydrolysis reactions, this is breaking down those macromolecules and releasing the energy that's stored in those bonds. So that energy can be used for something else, maybe used for a dehydration synthesis reaction. For drawing sort of a general picture of this, I'll draw just two sugar molecules here. Not going to draw the whole molecule, just parts of them. They have these OHs on them because sugars tend to have a lot of those. If I want to do dehydration synthesis and bond these together, what needs to happen is H2O needs to be removed. So water comes out, and I end up with those two molecules bonded together with an O in the middle. If I want to do hydrolysis, the opposite of that, so we could start out with these two sugars bonded together. We have to put water in, and we end up with the two sugar molecules separate. So I showed this with sugar molecules, but the same processes are going to happen when you're bonding fatty acids onto glycerols to make lipids, or when you're bonding two amino acids together to form a polypeptide. Hydrogen bonding occurs when we have hydrogen attached to a very electronegative element like oxygen or nitrogen, which results in the electrons getting pulled towards the more electronegative element, giving that part a kind of negative charge and this part a kind of positive charge. And then if we have two of these with this negative side and this positive side, they will tend to stick together because positives and negatives attract. Of course, this happens in water because we have oxygens that are very electronegative and hydrogens that are not. So we'll have the oxygen of one water molecule attracted to the hydrogen of another water molecule. We also call this the cohesion of water, so the water's sticking to each other. This creates surface tension for insects and whatnot to walk on. Water will also stick to other things. For example, the inside of a thin tube through hydrogen bonding, and we call that adhesion. This property also makes water tend to want to stick together and stay as a liquid, which is important for life on Earth. It also means that it can absorb a lot of energy without changing temperature. It takes a lot of energy put into that water to get it to move around a lot, which is what an increased temperature is, because the waters, again, want to stick to each other. So generally, if you're asked something about water and why does it have this property, most likely the answer is something to do with hydrogen bonding. Right. Polar and nonpolar molecules, we kind of talked about that with the hydrogen bonding, but polar molecules have an uneven distribution of charges / electrons. Which gives them a partial negative and a partial positive side. while polar molecules have a more even electron distribution, which means they don't really have a negative or positive side. They're kind of the same charge all the way around. The element that is present in most polar organic molecules is oxygen, because oxygen is very electronegative, and so it tends to pull those electrons to itself. Particularly if you have a smaller molecule with oxygen, you're definitely looking at something polar. Now, the fatty acids do have like a little oxygen over on the glycerol on this end, but they're so big, and all the rest of this stuff is just carbons and hydrogens, which don't really create polarity, that these are still nonpolar even though they have a little bit of oxygen. But if you've got a smaller molecule like a water that has an oxygen, that's going to make a pretty big pole, and that's going to give us a very polar molecule. Okay, capillary action is the tendency of waters to move up a thin tube, such as the inside of a root cell of a plant. The properties that contribute to it are the cohesion, because the water molecules will stick to each other, which can kind of pull them up in a chain, and adhesion, because the water molecules will also stick to the side of this tube, which will allow them to kind of climb up. Of course, water can only adhere to things that are polar molecules. Like water can't stick to wax paper, for example, because it's covered in wax, which is nonpolar. It doesn't adhere to that. So we can assume that the insides of these tubes are, in fact, polar. Water has a high specific heat capacity, and we already talked about this a little bit too, but again, that's the ability of water to absorb a lot of heat without changing temperature. It means water is very stable when it comes to temperature. It's hard for it to heat up. It's also hard for it to cool down. This is important for living organisms because we're full of water, which allows us to maintain a stable body temperature, and the oceans are full of water, which helps our planet to maintain a more stable temperature. Evaporative cooling basically means that as water evaporates off of a surface, it takes a lot of energy to transfer water from a liquid to a gas, and so that energy gets absorbed by the water molecules, allowing them to leave, resulting in whatever is left behind on the surface being cooler. So water cools us off as it evaporates. Why does ice float? Ice is less dense than water. Floating all has to do with density. So it's less dense than liquid water. And this is important because that whole idea that ponds or oceans or rivers, whatever, freeze from the top down, so that our little fish that are under this layer of ice, it can be slightly warmer below because the ice on the top will insulate it, allowing liquid water to remain down here for the animals that are down there. What kind of molecules does water dissolve? Water dissolves polar molecules and most ionic. Does not dissolve nonpolar molecules. If we have an ionic substance, draw an ionic substance with some positive ions and some negative ions, or a polar molecule with positive ends and negative ends, the negative oxygens in the water will surround the positive ions, and the positive hydrogens in the water will surround the negative ions, or the negative parts of polar molecules, and in that way it can break up and dissolve those molecules. Okay. On to proteins. There are 4 levels of protein structure. Primary structure is just the chain of amino acids. So this chain of amino acids, which is not folded up or anything, just the plain old polypeptide, That is the primary structure. Secondary structure is we start folding up that chain. So we get pleats and we get helixes. So we take bits of that chain and start like pleating them or spiraling them. That's hard to draw with this. Spiral. Something like that. Start folding up that chain again based on the R groups of that protein is going to determine how it folds up. Tertiary is we take this whole thing and we fold it up into a big protein. Again, this is going to depend on the R groups. Polar R groups are going to tend to stay to the outside because that's where the water is, and non-polar R groups are going to tend to go towards the inside because that's where it's not watery. And finally, quaternary structure is when we take multiple of these folded up proteins and put them together into one big clump. So some proteins only have up to tertiary structure, and some are made of multiple folded polypeptides. Enzyme activity versus temperature. Activity, temperature. Generally, as the temperature increases, the enzyme will start working better and better until it reaches its maximum efficiency, and then we get a big drop off. And we get this big drop off because the enzyme denatures, meaning it changes shape. It can no longer fit the substrate, so it doesn't work anymore. This is like scrambled eggs. Enzymes are proteins. Your eggs got scrambled. The protein is all tangled up now. It doesn't work anymore. Enzyme activity versus pH tends to be a little bit smoother of a curve, but we still get that denaturing at the extreme ends. And each enzyme has its own sort of ideal pH at which it functions. Enzyme activity versus substrate concentration. So we generally expect as we get more substrate in there, the enzyme is going to work better and better until it reaches the point of saturation. When all of the enzymes are working at maximum capacity, they're all as busy as they can be, and the reaction just can't go any faster no matter how much more substrate you add unless you add more enzymes. Why do enzyme reactions, like in our lab, inevitably slow down over time? So we have the product here, and we have time, product produced over time, so it's going to go, start making a lot of product, but eventually it's going to level off. It's going to stop making so much product. So what's happening here is the enzyme is running out of substrate. In the lab, there was only so much hydrogen peroxide in there for the enzyme to catalyze. So as the catalase breaks down the hydrogen peroxide, eventually it starts running out of hydrogen peroxide. And so it can't keep producing the product, the oxygen, at the same rate it was before. We can calculate the rate of enzyme activity based on a graph of product formed over time. And so when we're trying to find a rate, what that's really asking us to do with a graph or data table is to find the slope. So I need to figure out what my points are from 30 to 90 seconds. So 30 seconds, it looks like I'm at about, let's say, 0.01 milligrams of product, and at 90 seconds I'm at 0.04 milligrams of products. And so my formula for slope again is y2 minus y1 over x2 minus x1. So I'm going to say 0.04 minus 0.01 over 90 minus 30. Which gives me 0.0005 milligrams per second as my rate. Most common elements found in organic molecules, that is CHNOPs, where carbs contain CHO, lipids also contain only CHO, mostly this carbon hydrogen. Proteins contain CHNO and sometimes sulfur, but definitely have to contain nitrogen. And then nucleic acids, DNA we're not really doing, but those contain CHNOP, just FYI. The important thing to realize here, especially with the proteins, is that nitrogen is required to produce proteins. So if we draw an enzyme, example enzyme, let's say this is our enzyme and this is our substrate. Remember that the substrate has to fit perfectly into the active site of the enzyme. So if we have an inhibitor, let's say a competitive inhibitor, that's going to be a molecule that is similar shape to the substrate, but not quite the same, which is going to block the substrate from being able to get into the active site of the enzyme. A non-competitive inhibitor, here's our enzyme again, and let's say that this one actually has a little notch over here. So a non-competitive inhibitor is going to bind to a different site in the enzyme called the allosteric site. And when that happens, the enzyme's active site will again change shape, having the same sort of result that the substrate cannot bind in the active site. A saturated fat versus a unsaturated fat. So a saturated fatty acid is going to be shaped like this, basically, and it's going to have every carbon bonded to the maximum number of hydrogens, only single bonds in there. A unsaturated fat is going to have some double bonds in there, which form sort of kinks and bends in the chain. The general shape of each of these, if we have a saturated fat, a triglyceride will have 3 of these, and they'll stack really nicely on top of each other, which allows us to have sort of a solid. Unsaturated will have these bends, which will make it so they don't stack so nicely on top of each other, and so that is more likely to be a liquid. Okay, this got moved over a little bit, but the equation would be enzyme plus substrate forms the enzyme substrate complex, and then we end up with the enzyme and the products. So the enzyme doesn't get used up in the reaction. It's free to interact with new substrate once it has released the products. A few examples of lipids. Well, we have obviously the triglycerides we were talking about on the other page, which are long-term energy storage. They're very energy rich. They can be broken down into a lot of energy, and they can be stored as fat. These can also function as insulation, like blubber on seals or cushioning for your organs. We have waxes, for example, earwax or cholesterol. Which lubricates cell membranes. This would be sort of protection from things getting into your ear canals. We have the steroid hormones, which are chemical messengers. We have different oils, which can be used for waterproofing, like in otters and aquatic birds. So there's just a few different examples. Organic molecules usually have covalent bonds. And what is the importance of the specific sequence of amino acids in the function of a protein? So remember, shape is key. Proteins' functions depend on their shape. So if we have the wrong amino acid, it's not going to fold up the same way. It's going to make a different shape, and that protein probably won't work. So if we exchange, especially if we exchange like a polar amino acid with a non-polar amino acid, that can mess up the entire way this folds up, and it can totally mess up the shape. And again, the protein would not be able to function. Now we might be able to get away with exchanging a very similar amino acid with another similar amino acid, but in most cases, if there's a switch, if there's the wrong amino acid in there, then that protein just isn't going to be able to work because it's just not going to fold up right and it's not going to have the correct shape. Okay, so that's it for this review. I hope it was helpful. Please ask if you have any questions.